Namaste to all the students. Kushal Vidya, myself JB and Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. And in this session, we are going to discuss about nucleus structure, function, and extracellular matrix. And this session is related to cell biology course of program BTech FBT and BSc FST second sem. So, before starting this session, you have to understand that what are the basics and what are the basic discoveries related to the nucleus. So nucleus was discovered by an English biologist, which is Robert Brown, and he discovered the nucleus in 1831, and it is the important part of the cell and which exerting a controlling influence on all cell activities. It is usually spherical or oval structure, mostly located in the center of the cell. Some sieve tubes of vascular plants and the red blood cells of mammals, and they are like the nuclei at the maturity level. So means sieve tubes of the vascular plants and RBCs are not having any kind of a nucleus at their maturation level. If we are talking about the structure of the nucleus, so nucleus is a membrane-bound structure and double membranous bound structure that contains a cell hereditary information in the form of chromosomes and control the cell's growth and the reproduction. It is commonly the most prominent organelle in the cell, and the nucleus is surrounded by the structure called nuclear membrane. This membrane separates the content of the nucleus from the cytoplasm, and the cell's chromosomes are also housed within the nucleus. And the fluid which is filled up within the nucleus is known as nucleoplast. And chromosomes that contains the DNA which provides the genetic information which are necessary for the production of other cell components and for the reproduction of the life. So that's why nucleus is very much important for the cell and for the life. And if you are talking about the structure of the nucleus in detail, so average diameter of the nucleus is approximately 6 micrometer, which occupies around 10% of the cell volume. And if you are talking about the overall structures which are involved in the formation of the nucleus, so first one is the nuclear envelope, then nuclear membrane, nucleolus, chromosomes, and nucleoplast. It will come. First of all, we are going to discuss about the nuclear envelope. So nuclear envelope is double layered membrane which enclosing the nucleus of eukaryotic cells. This nuclear envelope has pores that allow the passage of material into and out of the nucleus, and it is also called nuclear membrane. A nuclear membrane, also known as nucleolemma or karyotheca, and it is the phospholipid bilayer membrane which surrounds the genetic material and nucleolus in the eukaryotic cell. The nuclear membrane consists of two lipid bilayer, the inner nuclear membrane and the outer nuclear membrane. Next important structure which comes up in the nucleus structure, so that one is the nucleolus. So nucleolus present within the nucleus, and this nucleolus takes up approximately 25% of the volume of nucleus, and this structure is made up of the protein and RNA. The main function of this nucleolus is to rewrite the ribosomal RNA, means rRNA, and combine it with proteins. This results in the formation of incomplete ribosomes. So somehow this nucleolus is involved in the formation of immature ribosomes. Next component of the nucleus is chromosomes. So the function of the chromosomes, and chromosomes are basically thread-like structures which are found in the nucleus of both animal and plant cells. They are made up of the protein and one molecule of deoxyribonucleic acid, means DNA. and proteins will be histone proteins in nature. And if you are talking about the next component, which is nucleoplast, just like the cytoplasm found inside a cell, nucleus contains nucleoplast, also known as karyoplast. The nucleoplasm is a type of protoplasm that is made up mostly of the water, a mixture of various molecules and dissolved ions. It is completely enclosed within the nuclear membrane or nuclear envelope. So, this is the overview of the structure of nucleus of cell. Here I am going to share the screen of diagram of nucleus, by which you can understand the cell nucleus structure more properly. This is a nuclear envelope which is having two membranes, outer membrane and the inner membrane. And this one is which are having nuclear pore by which material can come in and come out from the cell. And on the other hand, nucleolus will also be present within the cell, as I have already told you that it is involved in the immature ribosome formation, and these thread-like structures are known as chromatids, which will become condensed and are known as chromosome in cell cycle or cell division process. And the fluid which has been filled up within the nucleus is known as nucleoplast. So this is the basic structure of nucleus. And if you are talking about the work of or function of the nucleus, so here, first of all, as I have already told you, that nucleus is having hereditarily information in the form of the chromosomes, means chromosomes which are having proteins along with the DNA molecule. So DNA are having coding or non-coding regions, and they are having the genes which further express in the form of the RNA and further for the proteins. So somehow these chromosomes are involved in the transformation of the information from one generation to the second generation. And here you can understand this concept by this flow diagram. So you can simply see here, this is a nucleus structure which is having the DNA and along with the proteins and are making the chromatin thread-like structures. So this is the DNA molecule which is having coding region, exon and intron regions, means non-coding regions. So first of all, DNA can replicate itself and they can make the new DNA molecules, and that process is known as the replication process. And on the other hand, these are DNA molecule can synthesize the RNA molecules by the help of one process which is known as transcription process. In the transcription process, new immature or raw RNA molecules will be generated. These raw RNA molecules will have coding and non-coding regions both. After the completion of the raw materials of RNA, it will go for the RNA processing, and this RNA processing involve RNA splicing, capping, poly-A tail region, and some chemical modification happens. So after the completion of the RNA processing, mature RNA will be generated. Here in the diagram, you can see that this is the messenger RNA. This is mature messenger RNA, which is having only coding region, means exon region, and capping region at the 5' end, and poly-A tail region at the 3' end of the RNA. Now this messenger RNA will further transfer or further export to the cytoplasm of the cell, where this messenger RNA will be involved in the formation of proteins, means it will be involved in the protein synthesis, or you can say the process is translation. So it will involve in the translation process, and further it will form the protein molecules, and these proteins molecules are involved in further growth, structural, or it can be secretory, whatever their destination. So they will create like these proteins, and that's how nucleus do their work. They are storing the genetic materials, they are storing the genetic information, and after they're storing genetic information, they will go for the transcription. They will create the RNA molecules, and these RNA molecules further will go and further will transport to the cytoplasm, where it will be involved. They all are involved in the translation process, and they will synthesize the proteins, and these kind of proteins are involved in somehow in the secretion process, in some structural building maintenance. So that's how nucleus is involved in all kind of a work of a cell, and that's how they are involved in the cell growth and cell reproduction life. So this is the sum up of nucleus session. And if you are now come to the second topic and second session, which is the extracellular matrix. So most animal cell release material into the extracellular space, creating a complex meshwork of the protein and carbohydrates called the extracellular matrix, ECM. A major component of the extracellular matrix is the protein which is known as collagen. So if examiner is going to ask what is the major component of the extracellular matrix, then you have to give answer collagen. Collagen protein are modified with the carbohydrates, and once they are released from the cell, they assemble into long fibers called collagen fibrin. Collagen plays a key role in giving tissues strength and structural integrity. Human genetic disorders that affect collagen, such as uh, Ehlers-Danlos syndrome, results in fragile tissues that stretch and tear too easily. So that's how collagen play a role in giving tissues strength, and if it is not present in human body, so it will give this kind of a syndrome, and by which that person becomes so fragile, and they can't do stretching and some physical work. If they will do so, their tissue will easily tear. And in extracellular matrix, collagen fibers are interwoven with a class of carbohydrate bearing proteoglycans, which may be attached to a long polysaccharide backbone as shown in the picture. I will already show you in the picture in the next slide. So the extracellular matrix also contains many types of the proteins and the carbohydrate. You can understand extracellular matrix structure by this diagram. So here I'm sharing the screen of the slide. This is a plasma membrane, and this one is a cytoplasm, and this region is known as extracellular matrix, which is having collagen fibers, and somehow they are having fibronectin and proteoglycans. If we zoom on the proteoglycan complex, so you can simply see these are the carbohydrates, and they are attached to the protein molecules, and this one is a polysaccharide, and that's how this is a complex or conjugated structure of the proteoglycans. So extracellular matrix is having many kind of proteins, many kind of carbohydrates, in which collagen fiber is very much prominent, and second one is the proteoglycans, and somehow fibronectin and integrin and some microfilaments are also involved. The extracellular matrix is directly connected to the cells it surrounds. Some of the key connectors are proteins called integrins, which are embedded in the plasma membrane, and proteins in the extracellular matrix, like the fibronectin molecule shown in green in the diagram above, can act as bridges between integrin and other extracellular matrix proteins such as collagen. On the inner side of the membrane, the integrins are linked to the cytoskeleton. Integrin anchors the cell to the extracellular matrix. In addition, they help it sense its environment. They can detect both chemical and mechanical cues from the extracellular matrix and trigger signaling pathway. Blood clotting provides another example of communication between cells and the extracellular matrix. When the cell lining of blood vessel are damaged, they display a protein receptor called tissue factor. When tissue factor bind to a molecule present in the extracellular matrix, it triggers a range of responses that reduces blood loss. For instance, it causes platelets to stick to the wall of the damaged blood vessel and stimulate them to produce clotting factors. So that's how extracellular matrix involve in major events of the cell. And that's how now this session is sum up, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.